I am Chris Hound, VP of Preclinical Research, and at Gordian Biotechnology, we use in vivo mosaic screens to uncover novel gene therapies for complex, non-monogenic diseases. Here are my disclosures. So the last decades have seen breakthroughs in AAV. We have 8 AAV-approved therapies. Some of these are truly miraculous. We know the success story of Zolgensma. This has changed patients' and families' lives. Now, despite these successes and the field-wide advances in better capsids, scalable manufacturing, and reduced immunogenicity, despite all this hard work, we have seen a recent pullback the last year by pharma, driven by high costs, complexity, and narrow patient reach. And at Gordian, we have the vision to overcome this by applying AAV to common age-related diseases of millions, not just thousands, to unlock the economic scales needed for investment, manufacturing, and payer adoption. So how do we do this? Well, we need to know what gene to put into these miraculous vectors for non-monogenic diseases. And to do this, we use an in vivo pooled mosaic screening platform. Here we start with a library of AAV interventions. These have a GFP tag and a barcode. We then deliver these at a low transduction dose into the best available animal model. They're at this low dose such that the tissue remains largely unaffected and still in its disease state, with only sparse amounts of transduced neurons, transduced cells, potentially expressing a therapeutic gene. After 1 month, we then pull out the transduced cells, do FACS sorting, single-cell sequencing, and then transcriptomics to understand which of those therapeutic genes alter the cells in a favorable direction at the single-cell transcriptome level. After the screening process, we then go to the next stage of target or hit validation to see if the single-cell results were actually predictive of phenotypic change. Today I'll speak to 2 programs. The first is osteoarthritis. People know what this is. This is a massive growing population. Unfortunately, for those here, for those listening online, 1 in 2 of you, if you don't have it already, will develop OA in your life. There are no FDA-approved disease-modifying treatments. You have pain management or total knee replacement, okay? Nothing in the middle. This is a really attractive target for AAV. You need a very small dose required, so very small cost of goods. It's localized, has a beautiful safety profile. And for us, there is a terrific animal model, very similar to human disease, and that is retired racehorses that develop spontaneous OA, very similar to human, and you can see some histology here of an OA horse compared to OA patients. And we have access to these horses through these awesome collaborators at University of Florida. And this is how it works. We have our head of osteoarthritis, Niti Sahu, who really drove this program, is responsible for all the results I'm about to present, who created this first library of 45 targets. Since then, we've run many more libraries, but this is just the first one. The library composed of many different potentially therapeutic genes covering different classes and different potential mechanisms of action. We inject this into the fetlock joint of a 5 to 15-year-old osteoarthritic horse, and then 1 month later, harvest the joints, isolate the GFP-positive cells, and perform the single-cell transcriptomics. Okay, now this is my favorite slide. This will be new to everyone, so I'll take a bit of time going through this. This is a heat map of the therapeutic transcriptional change for all our interventions for various disease features, okay? Blue indicates that the intervention is making the cells for that disease feature become more healthy at the single cell level. Red is saying that it's going in the other direction. A disease feature on the y-axis is actually a curated set of genes chosen by our disease lead that correspond to an important disease process, such as cartilage matrix integrity, oxidative phosphorylation, apoptosis, etc. And to give an example of that, I'm going to unblind one of these, and this is AAV expressing FGF18. And FGF18 is actually an investigational new drug for osteoarthritis. It's as a recombinant protein that has known regenerative effects. And in our screen, it actually comes up, you see, as a very highly shifting the cells towards a cartilage matrix improvement. Once we saw these results, we were extremely excited because it meant the platform was working. And then, of course, we have all these other interventions that now we can take to the next level in the HIT validation phase. And I should point out that the FGF18 did okay in the trials. It didn't get approved because it had no effect on pain. So we need better treatments there. For our HIT validation for osteoarthritis, we have access to this awesome human ex vivo system. This is fresh OA tissue taken from people who have just had a total knee replacement. We co-culture the chondrocytes and the synovial cells. This is a gold standard ex vivo system. We add our AAV expressing the HITs, and then measure total glycosaminoglycan synthesis, which is indicative of cartilage health. You can see the control is at 5 micrograms per micrograms DNA. We have our negative control, an inflammatory cocktail, which reduces that, a positive control, recombinant FGF18, which increases GAG synthesis, and then 4 out of our 8 hits from that screen showing a significant increase in GAG synthesis. We then took the 2 best of these to lead status. We refer to these as OMEN 12 and OMEN 13. And in this system, we showed that they are truly disease modifying. In addition to increasing GAG synthesis, they also decrease levels of cartilage degrading enzymes, MMP12 and MMP13. And very importantly, they are also reduce inflammatory mediators such as GM, CSF, and there was a trend there for interleukin-1 beta. So now we've started in this great animal model, the horse, we have human tissue, and now let's go to the best rodent model that we believe for osteoarthritis. And this is 2-year-old black 6 mice, very precious, very expensive. It's a non-surgical model. They're just hard to come by. And what we do is inject now our OMEN 12 and OMEN 13 separately, bilateral injections into each mouse. Okay, so OMEN 12, obviously they're in the same animal. We do bilateral injections just because the mice are so precious and we want to increase the N, of course. 6 weeks later, harvest the tissue, do scoring, and what we have found is a significant reduction in the cartilage degeneration score of OMEN 12 and OMEN 13. Okay, so that is obviously really great. That is now human, horse, and mouse that we've seen that these novel therapies are having an effect. But pain is also a big component, and as I mentioned before, the investigational new drug, the recombinant FGF18, failed to get approved because it didn't have an effect on pain. So of course, we wanted to test how these interventions would do in a pain model. We have a decent model of pain. This is an inflammatory joint model. We inject monoiodoacetate into mice. This has a pretty short time course, so we have to inject the AAV prophylactically. These are unilateral injections, and the readout is a static weight-bearing assay. So we're forcing the animals to stand on their hind paws, and we're measuring the amounts of force that they put on each leg. So obviously, if you have an injury on one knee, they'll tend to put weight into the contralateral knee. And if you look at the data, at day 1, all the animals are putting equal amount of weight on the ipsilateral limb, but at day 7 and day 14, you have much more weight put on the contralateral. You can see for OMEN 12 and OMEN 13 at day 17, at day 7 and day 14, you do have a therapeutic effects closer to more even distribution. At day 14, it's much nicer. Our positive control, which is an anti-inflammatory drug, celecoxib, of course, is the best. And yeah, we're very excited by these results, and very encouraging to have both structure and pain across these different animal models. With that, I'm going to spend a few more slides going to one of our other indications, and that is heart failure with preserved ejection fraction. Fortunately, it's not affecting as many people in this room, but it is still a large and rising burden. There are a few treatment options available, and this is also a tractable tissue target. You will need higher doses than a simple joint injection, but there are multiple AAV clinical trials targeting the heart, multiple awesome capsid companies out there working to reduce doses and get cost of goods down. For this, we also have great animal models. We have two. We have the ZSF1 rats and two hill mice that mirror key human features, most importantly, diastolic dysfunction. For those who aren't familiar with HFpEF, this is a disease where the hearts can pump fine, it just can't fill with blood well enough. So it doesn't have the diastolic function working normally, and this is because of thick, stiff walls in the ventricles, and it's really a problem, okay? Now, I won't go into the details too much. I just wanted to show you that the ZSF1 is the species that we did the screen in. In this screen, we did 162 targets. This was driven by Naoto Muraoka, our head of cardiac biology. And from this, we tested a lot of hits, but today, for time, I will just show you the results from one of our leads. And this is Herald-7, which we have seen to improve heart failure in a HFpEF mouse model. So I love this. We're going from the rat to the mouse, because these aren't monogenic diseases. We don't know the gene. So here, if we can show that this target is working in multiple species, we get even more confidence that this could work in humans. In this model, these animals do have existing, we develop the existing heart failure, then inject the AAV after they have this diastolic dysfunction, measure echocardiography, biomarkers, invasive hemodynamics, and I'll just show you the final time point. This is two measures of filling pressure, right? So the increased filling pressure is a problem with diastolic dysfunction. And on the left, you have echocardiography measurement. On the right, you have the invasive hemodynamics, and you see that we have a therapeutic effect of Herald-7. And with that, yep, I'll wrap it up. We have now osteoarthritis, heart failure programs. We are also looking at pulmonary fibrosis and liver fibrosis, obesity. Some of these indications may be still well applicable to AAV, but if not, we can use our target discovery platform for other modalities. I'd like to thank the whole team, especially our collaborators at University of Florida. And with that, I'm happy to take questions. Thank you.